We just talked about the HMP shunt. What is the important function of the irreversible oxidative reactions of the HMP shunt? Yeah, it's to make plenty of NADPH molecules for the body. We have our enzyme glucose 6-phosphate dehydrogenase, and it's going to be involved in both the HMP shunt and the respiratory burst. It plays a key role in regenerating NADPHs. Let's say that we have a substance that's going to be harmful to the body, like hydrogen peroxide, for example. It can cause oxidative damage to the body if not dealt with. So what will our body do to protect itself? The body will use a reduced form of glutathione to neutralize this hydrogen peroxide and convert it into water. However, to keep high supplies of this reduced glutathione available, the body will need lots of NADPHs. Where did I say we got most of our NADPH molecules? Yeah, the HMP shunt. So if we are missing the key enzyme, glucose 6-phosphate dehydrogenase, from the HMP shunt, we cannot make enough NADPH molecules, and thus our body can be subjected to oxidative stresses. What's this? Yes, an almost guaranteed test question. The boards love the enzyme glucose 6-phosphate dehydrogenase. I'd pay close attention if I were you, at least for the next few minutes. A G6PD deficiency is actually the most common human enzyme defect, and it's X-linked recessive. One of the main problems in a G6PD deficiency is that this prevents the reduction of glutathione, which makes cells more susceptible to oxidative damage. We just talked about this. Under normal conditions, most patients with a G6PD deficiency might not have any symptoms. However, when they're subjected to oxidative stress due to things like inflammation or drugs like sulfonamides, primaquine, isoniazid, anything that causes some sort of stress on the body, this can lead to a hemolytic anemia, where their red blood cells can't handle this oxidative stress and thus they'll lyse. These patients should also not eat fava beans, since these fava beans contain high levels of oxidants. On test day, make sure that you focus on the information provided. If the question depicts a black individual who's taking a new medication and thus they develop this hemolytic anemia, you should immediately think G6PD deficiency. With so many negative side effects of this G6PD deficiency, are there any positives for having the disease? Well, the only silver lining here is that a G6PD deficiency provides some resistance to malaria, possibly because of the higher oxidative stress. How else could you be tested on G6PD deficiency? One way that the boards like to present this is by showing you a slide depicting bite cells. Bite cells, which you can see here at the arrows, are indicative of a G6PD deficiency, since red blood cells have these bites taken out of it. So why in the heck would phagocytes bite off part of the red blood cells? In normal individuals, the NADPH serves as a protective antioxidant role to the red blood cell. If you're not able to produce that NADPH because of this disease, well, you're not able to regenerate the reduced form of the glutathione, and thus you can't protect the cell from hydrogen peroxide damage. If you can't protect that cell, you get denatured hemoglobin that precipitates within the cell. This denatured hemoglobin is going to be called a Heinz body, which you can see over here on the right picture. So what you're going to get is that during times of stress, you're going to get these Heinz bodies. Now, let's draw in a phagocyte as a Pac-Man here. They're going to come in and recognize these Heinz bodies within the red blood cell, and they're going to bite off pieces of that cell and try and get rid of these Heinz bodies or accumulations. That's why you get these little chunks taken out. These phagocytes are going to come in and bite off part of the cell. So thus, if you see Heinz bodies, or if you see bite cells on test day, immediately think G6PD and an NADPH deficiency. Although other disorders like thalassemias and asplenic patients may also cause Heinz bodies, but you should have multiple things on your differential, and one of those should include a G6PD deficiency. Let's now talk about the disorders of fructose metabolism. How does fructose, which is a monosaccharide carbohydrate, fit into this grand picture of metabolism that we're trying to paint? Well, fructose metabolism is important because it gets converted into a substrate for glycolysis and energy production. As you can see, we start off with a fructose, and eventually we'll end up with a glyceraldehyde 3-phosphate, the same molecule that's in our glycolysis pathway. Normally we start off with a fructose and it gets converted into a fructose 1-phosphate by the enzyme fructokinase. This is usually found in the liver. Since we're dealing with a kinase enzyme, what we're going to be doing is we're taking a phosphate group off of an ATP and getting it converted to a fructose at the 1 position. So we have a fructose 1-phosphate. The enzyme aldolase B then converts that fructose 1-phosphate that we just made into a dihydroxyacetone phosphate and a glyceraldehyde. The enzyme triose kinase can then combine that glyceraldehyde with an ATP molecule to create a glyceraldehyde 3-phosphate, which is one of the substrates of glycolysis. This is a normal, healthy patient. Now, what happens when you're missing the key enzymes in this pathway? Essential fructosuria is an autosomal recessive defect in fructokinase enzyme. If you're missing fructokinase, what happens? Well, fructokinase is the enzyme that phosphorylates our fructose after it enters the cell, and it's this phosphorylation that prevents that fructose from crossing the cell membrane and leaving that cell. Therefore, without this enzyme, there's nothing that keeps the fructose actually trapped within the cell, so it can wash out into the urine. Therefore, this is going to be benign and asymptomatic. However, fructose intolerance is a much more serious autosomal recessive disorder of the enzyme aldolase B. Without aldolase B, we're going to have fructose 1-phosphate accumulate within the cell since it's already phosphorylated and it can't leave the cell. This is going to consume a lot of the available phosphate stores, which inhibits glycogenolysis and gluconeogenesis, two processes that require large amounts of phosphate to operate. Without glycogenolysis, the breakdown of stored glucose, or gluconeogenesis, the formation of new glucose, this fructose intolerance can cause hypoglycemia, or low blood glucose levels. You'll normally notice symptoms of fructose intolerance after the consumption of fruit, juice, or honey. Will you see large amounts of glucose on urine dipstick if a patient has fructose intolerance? Well, no, because the urine dipstick is sensitive for glucose only. In fructose intolerance, patients will have detectable amounts of reducing sugars in the urine, but the sugars are not glucose, so the urine dipstick will actually turn up as negative. In addition to hypoglycemia, patients will also have jaundice, cirrhosis, and vomiting due to the toxic accumulation of this fructose 1-phosphate within the liver. Treatment for this is to not eat fructose or sucrose, since remember sucrose is a disaccharide composed of both fructose and glucose. In general, disorders of galactose metabolism, which we're about to talk about next, is going to be a lot more serious than our fructose disorders. We should take a moment and emphasize what we just learned. Is essential fructosuria or fructose intolerance more symptomatic, and why? Well, fructose intolerance is worse since a deficiency in aldolase B leads to a fructose 1-phosphate accumulation within the cell, reducing the available phosphate stores. This inhibits glycogenolysis and gluconeogenesis, leading to hypoglycemia. And then now we'll talk about the disorders of galactose metabolism. In galactose metabolism, galactose is converted into a glucose so that it can be used in glycolysis from the beginning of the pathway. This mostly takes place in the liver, but other tissues also metabolize galactose to a lesser extent. Normally, dietary lactose is broken down into both galactose and glucose. Next, the galactose is then phosphorylated by the enzyme galactokinase to produce galactose 1-phosphate. So again, we have another kinase enzyme, and all we're doing is we're attaching a phosphate at the 1 position. So then this galactose 1-phosphate is converted by uridyl transferase into a glucose 1-phosphate. This then can be converted into a glucose 6-phosphate, like I said, can enter in the glycolysis pathway from the beginning, so it can either be used for glycolysis or it could be used to finish the gluconeogenesis, depending on the energy needs of the body at the time. What happens if you're missing some of the galactose metabolic enzymes? Well, a galactokinase deficiency is an autosomal recessive deficiency of the enzyme galactokinase. This prevents the phosphorylation of our galactose, and instead it causes it to be converted into an alcohol form, or galactitol. This is catalyzed by the enzyme aldose reductase. The same enzyme which also converted glucose into sorbitol, which we'll see on the next slide. A galactokinase deficiency is usually a mild condition, but galactitol does accumulate within cells, and it can cause galactosemia, infantile cataracts, and failure to track objects or develop a social smile. So it's not benign completely, but it's not as bad as our next deficiency. Classic galactosemia is a more serious autosomal recessive deficiency of the galactose 1 phosphate uridyl transferase enzyme. A deficiency of this galactose 1 phosphate uridyl transferase is going to cause galactose 1 phosphate to accumulate in cells. It'll also cause this galactitol to accumulate in cells. So it's going to cause some of the same effects as galactokinase deficiency, such as the infantile cataracts, but as well we're going to get some new symptoms due to that galactose 1 phosphate accumulation, such as jaundice, hepatomegaly, or E. coli sepsis in neonates. These are similar to what you would see in fructose intolerance, since in both of the cases you're accumulating a phosphorylated monosaccharide inside the liver cells, inside those hepatocytes, and actually causing liver damage, and you're losing your phosphate stores in the process. The treatment for classic galactosemia is simple. You just don't eat anything with either galactose or lactose in it, which is the disaccharide made up of galactose and glucose. As promised, we'll talk about sorbitol. Sorbitol, which you can see up here, is basically a reduced alcohol form of glucose. Sorbitol is going to be formed by the enzyme aldose reductase. Where did we see aldose reductase before? Oh yeah, in the conversion on the last slide of galactose into galactitol. So our sorbitol is going to be created by aldose reductase. So we have a molecule of glucose, it gets converted to sorbitol, where it gets trapped inside of cells. And it can then be later converted into a form of fructose by sorbitol dehydrogenase. Not all cells in the body have this enzyme sorbitol dehydrogenase. So some lack sorbitol dehydrogenase and some have it. So fortunately, our liver, ovaries, and seminal vesicles all contain both enzymes aldose reductase and sorbitol dehydrogenase. However, if a tissue lacks the sorbitol dehydrogenase enzyme, so we actually get this crossed out, we're going to have toxic levels of sorbitol accumulate. So we're not able to convert that sorbitol into fructose. So we're going to get this sorbitol trapped within the cell, which can cause damage because this is going to be osmotically active. What do I mean when I say something is osmotically active? High amounts of sorbitol can create a concentration gradient that pulls water into the cell, causing it to swell. In states of persistent hyperglycemia, such as diabetes, high sorbitol levels can cause damage to the lens, the retina, the kidneys, and the Schwann cells, since all of these have only aldose reductase present and not actually the sorbitol dehydrogenase that'll get rid of this sorbitol within the cell. This can cause cataracts, retinopathy, glomerular disease, and peripheral neuropathy, all of which are found in the disease diabetes. So let's turn this into a question. A poorly controlled type 2 diabetic man has poor vision and tingling in his feet. How may sorbitol be at fault? Well, this is because chronic hyperglycemia or high blood sugar levels causes sorbitol to accumulate in these organs and cause osmotic damage to cells. This is most prominent in cells that lack sorbitol dehydrogenase enzymes to break down that sorbitol that gets trapped in the cell. So this will include our Schwann cells, the lens, the retina, and the kidney. Have you ever wondered what someone meant when they said that they're lactose intolerant? Well, lactase is a brush border enzyme found in the small intestine which hydrolyzes or splits the disaccharide lactose into its constituents, glucose and galactose, which can then be brought into an enterocyte. A deficiency in this enzyme can be either primary or secondary. So a primary deficiency is due to aging or losing expression of the lactase allele, usually seen in Asian people, Africans, and Native American descent. Lactase deficiency can also be secondary, which usually is transient following a bout of gastroenteritis or from an autoimmune disease. Lactase deficiency can also be congenital, but this is usually rare. So how does a lactase deficiency typically present? Well, symptoms of lactase deficiency and subsequent lactose intolerance include bloating, cramps, and osmotic diarrhea. You'll see these symptoms because lactose will not be absorbed into the cells and therefore will pull water into the GI tract due to this osmotic gradient. So water will follow the osmotic gradient that this lactose is going to create. Bacteria in the gut can then use this lactose to form methane, leading to the flatulence. You can diagnose lactase deficiency clinically, but stool testing will demonstrate a decreased pH and breath tests will show an increase in the hydrogen content. Let's say you're attending orders an intestinal biopsy to determine the cause of this patient's severe, persistent diarrhea and cramping. What are the biopsy results going to show if they have lactase deficiency? Well, hopefully you're thinking that the biopsy results will be normal. Remember that lactase is a brush border enzyme. If it's deficient, there will be a lack of enzymatic activity at the brush border, but the GI epithelium will still look normal under a microscope. Microscopes are not going to see enzyme deficiencies. That means that the biopsy will appear normal, so the treatment for lactase deficiency is going to include avoiding lactose in the diet or taking these lactase pills along with food. So far, we've covered the concept of carbohydrate metabolism. Now we'll move into the realm of proteins and their building blocks, the amino acids. Only the L form of amino acids are going to be found in proteins. As you might remember from undergraduate biochemistry class, the amino acids can be categorized by whether they're essential or non-essential and into whether they're acidic, basic, or neutral. The essential amino acids have to be supplied by the diet since humans cannot produce them on their own. The glucogenic essential amino acids, which simply means they have to be converted to glucose via gluconeogenesis in the liver, are methionine, valine, and histidine. The essential amino acids that can be either glucogenic or ketogenic include the isoleucines, phenylalanine, threonine, and tryptophans. As a side note, I did throw in the structures to these molecules. These are not going to be important for test day. However, you do need to know how to classify each one of these, but the structures you don't have to memorize. Alright, and do you remember what the purely ketogenic amino acids are? The purely ketogenic essential amino acids, which can be converted to ketone bodies, are your leucine and your lysine. And now do you remember what enzyme deficiency we talked about earlier you would have to give purely ketogenic amino acids as a treatment? What enzyme deficiency would you have if you gave purely ketogenic essential amino acids as a treatment? Well, hopefully you remember pyruvate dehydrogenase deficiency, which requires giving these ketogenic amino acids, the leucine and the lysine, to bypass the deficient or the defective enzyme pyruvate dehydrogenase. Also, some amino acids are considered to be partially essential because we can synthesize them in our bodies, but sometimes we can't synthesize them fast enough to meet metabolic demands. So we'll need a little extra help from our diet. In particular, arginine is going to be considered a partially essential amino acid. So it's not required by adults, but kids cannot produce the amino acid arginine fast enough to meet up with metabolic growth demands. So in this case, arginine would be required in the diet. Now just remember that all other amino acids I didn't cover will be your non-essential amino acids. We also have acidic amino acids, which have a negative charge at physiologic pH. There are only two acidic amino acids, which includes aspartate and glutamate. The alternate name for these will be your aspartic acid and glutamic acid. The basic amino acids, which are usually positively charged at physiologic pH, are going to include your arginine, lysine, and histidine. Arginine is the most basic and histidine doesn't actually have a positive charge at physiologic pH. We can incorporate our discussion earlier about histones, the molecules that DNA wraps around. Remember that DNA is negatively charged, so the histones can increase their composition of positively charged or basic amino acids to attract and hold on to DNA. Interestingly, the only basic amino acid that sounds like histone, which is histidine, is the only basic amino acid that is not used for this purpose. So you're not really going to find histidine in a histone.